Hello and welcome. This is Dr. DeMayo. I'm here to talk about the second part of your AHS 131 lecture, and I'm calling this part of Lab 1. So this is the beginning of Lab 1, and it's also the end of your Lecture 1. So Lab 1 is going to cover the development of the somite and the parts of the somite. And we're starting from 2 cells, sperm and egg, and moving along. And then in your book, you're going to be doing exercises 1 through 8 in a lab book, and I will help you through that live as we go through it tomorrow or Sunday morning. So let's move along here. So the first thing is, how do you define a stem cell? A stem cell is an undifferentiated cell that gives rise to more cells of the same type or cells of specific type. A good example of stem cells within all the different tissues of the body, the stem cell for all the connective tissues of the body comes from embryonic mesenchyme. That's a name for the embryonic connective tissue. And a zygote is the first stem cell of the body. All cells of the body start here. Each cell that arises from this point are related to the zygote. And as we discuss these next steps, keep the following statement in mind. We talked about this Saturday in our lecture, that each and every cell in your body are related to each other. So day 1, syngamy means it's when the 2 gametes come together. A gamete is a sex cell, and the male, you see on the left, the blue sperm, that's a gamete, the male gamete, and in the big purple in the middle is the woman's egg, that's her gamete. So you have male and female gametes coming together to form one cell called a zygote. That zygote is actually your first stem cell of the human body. And then day 2, you end up getting 4 cells. They're undifferentiated. What does it mean to be undifferentiated? Undifferentiated means this: you cannot tell one cell from the other. So if you could click and drag each one of those cells out and look at it under your desktop and blow it up and magnify it, they look exactly the same. There's nothing different from each cell. So they're undifferentiated. And as you'll see, they start to differentiate and form different parts of the development, and then we start forming the human body. So day 4, we have this ball of undifferentiated cells. That's called the morula stage. Then it moves into the blastocyst. Now, if you notice this, what happens on the right, you see the fertilization occurs. On the right top there, it says sperm and the nucleus gets in. Many different sperm have to be working on it, and only one gets capacitated. And capacitation is a big word. You may have heard it before. But capacitation is when the cell actually, the female chemicals in the canal can actually stimulate and change the sperm cell so that those chemicals become more attracted to the oocyte. In other words, capacitation, to define it, is the changes that the sperm undergoes within the female reproductive tract. So the female reproductive tract can secrete chemicals that can not only enhance the swimming of the sperm by causing its flagella, it has little flagella, to beat faster and stronger, but also it can remove this thing called the MHC protein that identifies a different human body cell other than its own. So that MHC protein, that glycoprotein on the cell membrane of the sperm cell, is really an enemy. So what happens is the woman is going to selectively pick one through these chemicals, and that sperm will be allowed now to enter the cell. It's no longer an enemy. In other words, the oocyte will allow it in because it doesn't have that protein on it anymore. I think that's pretty amazing how the body does that. So after fertilization, you go to your zygote, and then you go to the 4-cell stage, and you go to the morula stage, all the way to the left, number 4. And then the morula stage is going to form into a blastocyst, and that's when the blastocyst is going to start to change from just 2 layers. It's going to have 3 layers with a middle layer in between, and then that is actually going to burrow its way into the uterine wall and implant. So days 6 to 9 are actually implantation within the uterus. So fertilization occurs where? Fertilization occurs in the fallopian tube. Or another name for the fallopian tube is what? The oviduct. So fertilization occurs in the fallopian tube, and then that fertilized egg is now going to become a zygote. It's going to go into morula, and then a blastula stage, and then the blastocyst is going to implant into the uterus. You see on the chart in the middle there, it shows you days 5, 6, 7, 8, 9, and you see it starting to move from day 3 down, 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 and gets into the uterine wall and then burrows its way in there. And that's called implantation. And then by day 15, the stage called gastrulation causes, gastrulation, and you get a rearrangement and a migration of cells, converting a 2-layered cell disc to 3. I said the blastocyst has 3 layers. I'm mistaken. It actually only has 2 layers. And the gastrulation stage has 3 layers. And here you can see your 3 layers: ectoderm, endoderm. In this picture, it doesn't show you mesoderm, but in this one, you can see here's the on the left, you see the blastocyst coming in, getting implanted, starts to get thicker and bigger, becomes a bilaminar embryo disc, and then it goes into the gastrulation stage and it becomes trilaminar, meaning you have 3 layers: ectoderm, mesoderm, and endoderm. Now they're important because we're going to go through this and you're going to see what each part will become in the body. So ectoderm is outside, mesoderm is in the middle, endoderm is inside deep. The ectoderm forms the following tissue: the epithelium and epidermis of the skin, and the entire nervous system. Can you imagine that? And then the next level, the mesoderm, the middle, forming the following tissues: connective tissue. Now mesenchyme is the tissue in the embryo that's all the connective tissue, and this will include the bones and the connective tissue of the peritoneum. And then the peritoneum is inside your abdomen. We're going to learn about those membranes in our lab, about the membranes surrounding the abdomen. And then you have muscle. That's the next thing that forms. Endoderm is the deepest layer, and that's forming the following tissues: the epithelium that lines the GI tract and lines the respiratory tract and the organs. So everything has a starting point. Day 15, the mesodermal cells migrate toward the cephalic end of the head of the embryo and forms the notochord. The notochord is actually the primitive disc, so to speak, and vertebrae. So the notochord is a hollow tube that helps eventually form the vertebral bodies, a portion of the vertebrae, and the nucleus pulposus of the disc. And that's the part that herniates out of the disc, the nucleus pulposus. And then the notochord, you see as an image, if you look at this image to the left, you're looking at a developing embryo, so to speak, and you can see the tail end and the head end. Notice at the head end, you see the notochord, and then you can see this primitive streak, which is going to be the neural tube. And then you see a different section. You see 2 different types of sections. The first one is like a longitudinal or a frontal section, or a coronal section, like you're cutting a hot dog straight down from the top all the way down, separating both sides as if you were going to put something in the middle of the hot dog. And then to the right, you're seeing a cross section as if you're chopping it into little pieces. And so in the center, you see the actual developing notochord right in the center. The amniotic cavity is up there, and then the yolk sac below. So now the next stage is neurulation from days 17 to 22. Now if you look in the chart on the left, it tells you neurulation, and the notochord induces overlying ectoderm to develop into the neural plate. So that's what this is all about. It's about forming the nerves and the brain and the spinal cord. But it's primitive at this point. First it forms a neural plate, then it folds, it forms neural folds, and the neural folds form the neural tube. And you can see these stages all the way across: neural groove, neural fold, neural tubes. By day 23 it becomes like a neural tube. And then in the bottom in the center, it shows you a cross section through that, and you can see the somite, which is formed from the mesoderm, And the somite has the three parts, the sclerotome, myotome, and dermatome. So neurulation is the process by which the neural plate, neural folds, and the neural tube forms. The neural crest cells, wow, typo, give rise to all sensory neurons. Postganglionic neurons of the peripheral nervous system, PNS means peripheral nervous system, and gives rise to the adrenal medulla cells, melanocytes, which are pigmented cells, the arachnoid and P of matter, which is part of the meninges covering the brain and spinal cord, and almost all the, blows my mind, almost all the connective tissue and skeletal components of the head. So your brain, you know, the nervous system, the brain and the spinal cord are all related to the bones of your skull. It's quite amazing if you think about it. And then the fourth week, we form these vesicles. So now this was just a tube, and the tube is starting to create these out pockets and thickenings, and you form these three primary vesicles, the prosencephalon, which will be the forebrain, the mesencephalon, which will be the midbrain, and the rhombencephalon, which will be the hindbrain. So the prosencephalon becomes a secondary vesicles, which would be the telencephalon and the diencephalon, and that is the cerebral hemispheres, and will also be the center of the cerebral cortex in the center, the basal ganglia and the thalamus and all that stuff. And so then the diencephalon is actually going to be where the thalamus and the hypothalamus is. So you have this prosencephalon forms into the telencephalon and then the diencephalon. And you can see the telencephalon will be the cerebral cortex, and then you see the eyes coming from that as well below there, and the thalamus, the hypothalamus, and the epithalamus, which is right in the center of the higher brain. And then the mesencephalon, the midbrain, will form the midbrain, of course, and the metencephalon forms the pons and cerebellum, and then the myencephalon forms the medulla oblongata. Then it shows you a breakdown, the fifth week, prosencephalon forming those different things we just went over. You could take some time to go over this on your own. Then the area adjacent to the myencephalon develop into the spinal cord. So you had this developing tube surrounded by somites. Watch, we're going to see that. So the next thing that we see is that the walls of the brain, let's look at those vesicles that we were talking about, the primary vesicles and the secondary vesicles. They will form, the walls of it will develop into the nerve tissue of the brain, and then the hollow tubes become the ventricles of the brain, and that's where the cerebral spinal fluid is produced and stored and flows and gets filled with it. The neural crest cells expand and form the protective structures of the brain. So most of the bones of the skull and the associated connective tissues and the meninges would be formed from neural crest cells. In day 17, the mesoderm adjacent to the developing neural tube differentiate into three layers, and they call that the paraxial mesoderm, and it develops into the somites. If you look on this picture, you see the ectoderm on the outside, the neural crest cells, the neural tube, and then you see the notochord, and then look, dermatome, myotome, and sclerotome. This is a cross section for this developing tube. So you cut it in half, like you're cutting a hot dog in half, right? Cross-section wise or transversely. And this is an image to show you a little bit about how the somites grow out and become these different parts of your body. So you see the brain and the skull forming here. There's the eye forming there. This is the pharyngeal pouches, which would be your throat area. And then this is the heart developing in another area close to the same area here, which will actually form the arms and the shoulders and the head and neck. And then this will form the mid-back, and then the organs will be forming in here. Oh, you can't see my pointer, I'm sorry. So you look at the heart bulge, and then you look at the arm bud, and from the arm bud, you go down even further, you see the somites go, and they call it the tail, but just before you get to the tail, you see the leg bud. So the legs are going to grow out from the pelvic area and grow down, and the somites are going to travel with it, the material in the somites, dermatome, myotome. And so this is a theory of the development of the species, in that when you watch the development of all the different embryos of all the different species, Hankel's phrase that ontogeny recapitulates phylogeny, it's a mouthful. It's a historical hypothesis that was never completely proven, but there is a pattern that you can definitely see where the development of the embryo of animals from fertilization to hatching or gestation, they go through stages that are similar to each other. And we just saw a picture that showed a tail, right? It said a tail, calling it a tail. And what happens is, and pharyngeal pouches, some people say that's similar to the gill slits on fish. And it is. But it doesn't mean that evolution is proven by that. What it does show is a definite pattern in the development of all the creatures on this planet. And each creature has a developmental pattern, and the human being is the highest on the order of all the phylum. And so human beings go through all the different stages of other animals. Other animals don't go to the levels that we have. So it's quite fascinating. I look at it as like a dressmaker. My mother her mother was a seamstress, and she used to make her own dresses, and she would get a pattern for the dress, right? So there's different patterns. There's a basic dress pattern, and then you can make it different in all different ways, but still there's a basic dress pattern. It's got to cover you, right? And so there's a basic biological systems, and then you can have these different modifications and changes and make it better or whatever. So it's just a pattern. And there are 42 to 44 pairs of somites by the 5th week. And each somite has 3 parts. And we talked about this. Dermatome is the spinal nerve and the skin it supplies. Myotome is the spinal nerve and the muscle it supplies. And sclerotome is the spinal nerve and the bone it supplies. So this is showing a dermatome chart of the body. And what you're seeing here is each level of the spine is actually related because there were somites surrounding each level, and from that somite it grew out to supply and grow the skin at that area. So notice that C2 is around the neck. The face is blank because on the face it's actually the trigeminal nerve, cranial nerve 5, that's supplying the skin of the face for sensory. But on the back of the head you see C2, C3, C4, C5 going down the arms, in the chest and the back, and the hips and the legs with the L1, L2, and so on and so forth. And this is the pattern of skin developing with its nerve supply. And then the myotome chart, this is just the best one I could find an image for you, is showing you that the muscles that move these different movements, cervical flexion, bending your chin towards your chest, that's the nerve supply mostly by C1 and C2. Extending your head back would be it doesn't show it there, but you have cervical side flexion as C3, elevating your scapula C4, and so on. Knee flexion S2, knee extension L3. So those levels of the spine have nerves that supply the muscles that do that. And there is a thing called Hiltons Law. And it's a great thing to do because you can help you to understand this a little simpler. Hilton's Law basically is a principle that says the nerves that supply a joint, let's say an elbow, also supplies the muscles that move that joint or the elbow. And it also supplies the skin covering it. So if you know the dermatomes that cover that joint, you can basically guess what the myotomes will be and what the sclerotome would be. Because the sclerotome is the spinal nerve that supplies what? The bone. It's the spinal nerve and the bone it supplies. And this is a chart showing different pain patterns from bones being irritated, the joints and the bones, especially in the spine. And so you see C3 referring there. This is a diffuse type of pain. It could be further deeper if they show it, you can have a more increased pain. But basically, you have different types of pain. You have nerve pain from the dermatome. You can have muscle weakness from a myotome. And then you can have joint pain that's kind of diffuse. It's hard to make it out, but it's from a joint being irritated referring pain, believe it or not.